Well, okay, let's go ahead and get started here. And as you can see, I did forget to open up those other tests, and I apologize. I have no excuses. I just forgot to do it. And I will go ahead and open them up as soon as we get out of class today. So now we're going to talk about the other half of the special senses. We're going to talk about the structures of the ear and how hearing works. And then we're going to take a detailed look at the eye and the brain. So, and we'll be looking at examples of brains that have been cut and opened up, and eyeballs that have been cut and opened up. I'm sorry, yeah, brains and eyeballs. So, okay. So anybody else have any questions on anything? Okay, then let's go ahead and get into special senses. And while this is a very busy slide, busier than I generally like, it does pack a lot of information into one place. The way we hear depends on pressure waves. We call them sound waves. They're really pressure waves moving through the air that enter through our outer ear, what we call the pinna, and they're collected by the outer ear and focused into our ear canal, or the auditory canal. And these pressure waves move down the canal and reach the tympanic membrane, striking it, we call it the eardrum, and causing it to vibrate. The vibrations of the tympanic membrane are then transferred to the middle ear, where we have three very small bones, what we call the hammer, the anvil, and the stirrup, or the incus, the malleus, and the stapes, that vibrate. So we are converting the pressure waves into mechanical waves, vibrations. And those vibrations travel through the three bones of the middle ear, and the stapes delivers those vibrations into the inner ear in a structure that looks very much like a snail. It's into a structure called the cochlea, and the cochlea is lined with tiny hairs inside in that swirl, that curled area there. That area of the cochlea is lined with hairs and fluid, just like it looks right there. Now, as those vibrations from the cochlea from the stapes are passed into the cochlea, the vibrations cause those tiny hairs, those very delicate hairs, to vibrate. And when they vibrate, they activate mechanoreceptors that, if they generate an action potential, send a signal to the auditory nerve to the brain, and the brain interprets what the sound is. So it's all about getting the hair cells inside the cochlea to vibrate. And if they vibrate, they have to be able to reach threshold so that they can generate an action potential that can then be interpreted. Because again, the ear has no idea what we are hearing, because it's just receiving vibrations, just receiving pressure waves turned into vibrations, and that's all. The brain has to interpret what those pressure waves mean. The pressure waves are turned into vibrations so that we can generate an action potential, but the brain itself, usually in the temporal lobe on either side of just below the parietal lobe, which is underneath the parietal bones, and the temporal lobe is underneath the temporal bones, then we can interpret what those vibrations actually mean. So the parts of the ear. The three big parts of the ear are the outer ear, the pinna, which is the portion that is visible on the outside, what we call, well, your ears, we have a big ear or a small ear, your lobes are attached or they're dangly. That's the outer ear, the pinna. We have an external auditory canal that leads into the middle ear. The middle ear starts at the tympanic membrane. We have glands there that generate a lot of wax, but the wax is there for a purpose. The wax is there to trap dust and dirt and bacteria and bugs and anything else that might get in there. And then on the other side of the tympanic membrane, we have the bones of the middle ear, the malleus, the incus, and the stapes, the hammer, the anvil, and the stirrup, if you will. There is an auditory tube that leads into the middle ear that opens up to relieve pressure. You know, when our ears pop, you know, the auditory tube goes from our nasal cavity into our middle ear. And it is an equalizer so that the pressure inside the middle ear is always the same as the pressure outside around us. So, and then our inner ear is made up of the cochlea, which leads to the auditory nerve. The cochlea is a structure that's shaped like a snail, and the auditory nerve comes off the cochlea. This is the cochlea is where these delicate hairs are that vibrate. And if we generate action potentials, they're going to go down the auditory nerve. Also in the inner ear, we have what are known as the semicircular canals. And these are three bony canals filled with fluid, lined with hair cells, just like the cochlea is, that are the semicircular canals run in three different planes, three different directions. And we determine our position in space as far as equilibrium and balance go from the direction that the hair cells are being tilted inside the semicircular canals. So here's our ear. There's the auricle or the pinna right there. This is the external auditory canal. The tympanic membrane is right there. The malleus, the incus, and the stirrup or stapes. The stapes connects to the inner ear through a structure known as the oval window. The oval window leads into a structure called the vestibule. And the vestibule is the area between the semicircular canals and the cochlea. And so the vibrations for for sound are admitted into the inner ear at the oval window, and then they pass into the cochlea and stimulate, hopefully, maybe, the inner ears, the hair cells inside the cochlea in the inner ear. Now, balance and equilibrium are based on the movement of those same types of hair cells inside these canals. And they send a signal to the brain through the vestibulocochlear nerve, balance and equilibrium. So now we take a good look at the middle ear behind the tympanic membrane. We can see there's the tympanic membrane there. There's the ear canal right here, auditory, external auditory canal. The malleus, it's taking the vibrations from the tympanic membrane. It vibrates. The incus receives those vibrations and passes it off to the stapes right here. And so the oval window is on the other side of the end of the stapes right there. And of course, there's the vestibule there, and there's the cochlea, and there's the semicircular canals. Now, this is the middle ear. You know, these bones vibrate. They vibrate from the pressure waves that cause the tympanic membrane to vibrate. And then we pass those vibrations on to the inner ear so that we can possibly generate an action potential from the vibrations. And then our brain can interpret what those action potentials mean. Now, a person that has conduction deafness would likely be unable to transfer the vibrations effectively from the tympanic membrane to the inner ear. And so some sort of device, perhaps a cochlear implant, perhaps a hearing aid of some sort, would allow those vibrations to be received by the cochlea and stimulate the auditory nerve. So that's conduction deafness can be helped with a cochlear implant. It can be helped with a hearing aid. Nerve deafness, damage to the auditory nerve, cannot be helped by either one. With conduction deafness, it means we're having a problem getting the signal to the cochlea. With nerve deafness, it means that we're having a problem getting the signal to the brain. Neither a cochlear implant nor a hearing aid can help nerve deafness. So here's our model of the ear. And you can see the various parts of the ear located here. There, for example, is the pinna, the auditory canal, the external auditory canal, the tympanic membrane, the ossicles, let me get this out of the way. This is what we call, this is the pharyngotympanic tube or eustachian tube, or the auditory tube, or auditory, whatever. This is the tube that opens up in the back wall of the nasal cavity to equalize air pressure in the middle ear. This is when you say your ears pop, because that tube, the old eustachian tube or the pharyngotympanic tube or the auditory tube, is simply there to allow pressure to be equalized. Because this is, the middle ear is a sealed chamber, pretty much. But let's equalize the pressure, because when the pressure rises in there, it'll hurt. And so we always enjoy having our, when our ears pop, we feel better. Okay. It's also a good passageway for a nasal infection to spread directly to the middle ear. It's a perfect pathway for some sort of pathogenic virus or pathogenic bacteria to migrate itself into the ear and cause an ear infection. Okay, there's the cochlea. There are the semicircular canals right there. Here's a top view looking down onto, we can see there's the tympanic membrane. There are the ossicles, the auditory nerve. There is the, again, the semicircular canals. There's the vestibule. The oval window is located right here at the vestibule so that vibrations come in to the inner ear through the oval window and are transferred over to the cochlea here. Oops, let me get back here. To the cochlea here. The semicircular canals will generate action potentials on their own without having to have a signal coming in. They depend on the movement of the head to distort those hair cells, so that we can tell which way we're moving. There's our internal ear. There's the cochlea right there. There is the stopes right there. The oval window is right behind the stopes. Here, this whole structure here is called the vestibule. And here are the three semicircular canals. Here's the cochlea over here. On the model, it looks like this. There's the stopes butted right up against the oval window. The cochlea, the stopes, the vestibule, And so now let's take a look at our model here. Let me get out of here for a second. And we've got a very large ear model here we get to use. You can see the size of the pinna is located right here. And it's designed to collect sound waves so that they move into this external auditory canal over here. And the idea behind the way the pinna works is simply it's a large enough funnel to trap sound waves to bring into the inner ear. The very first hearing aids worked on that same principle as the pinna. The very first hearing aid was someone who cupped their hand and put their hand behind the pinna so they could catch more sound waves. Later, large horns were used, large metal or ivory or wooden horns were carved to stick in the ear so that they could collect more sound waves. Those are some of the earliest devices used to assist in hearing. Once the pressure waves strike the tympanic membrane here, tympanic membrane vibrates, and we have then our ossicles transfer that vibration, and the vibration is delivered then to the vestibule of the inner ear by means of the stoppes. The stoppes butts up against the oval window right here. Now, this area that receives the vibration is the vestibule. Over here is the cochlea. You can see what the cochlea looks very much like a snail. You can see it. Let's take a closer look at it. There's the stoppes there. The oval window is right there. And here's the cochlea here. So the vibrations that are transferred to the inner ear go from the oval window into this area in here where we have the hair cells. Now the movement on equilibrium is based on hair cells inside these semicircular canals. You know that they're arrayed in 3 different planes. So vertical, horizontal, and vertical in two different directions, and then horizontal. So we can tell exactly where we're going or what's happening to us based on the movement of the hairs inside the canals. So there is no linkage between the get in there. There we go. Got it. There is no linkage between the semicircular canals and the cochlea, other than they're sharing the structure known as the vestibule. Now, behind the cochlea, we have the auditory nerve and the balance and equilibrium, the vestibulocochlear nerve. They're sharing that structure together. So now you'll notice that all of this bone in here is all spongy bone, surrounding the ear. And this works out very well for a cochlear implant, because since this is spongy bone, sound waves can be transferred very easily through this bone. So a cochlear implant usually lost my inner ear again. Cochlear implant usually involves something near the mastoid process, or in that area, slightly above the mastoid process, maybe in here, to take advantage of this spongy bone, because with a cochlear implant, sound waves can travel through the bone to the inner ear. So we don't have to, this is a different type of hearing aid, if you will, because we're bypassing the external ear, we're bypassing the middle ear, we're going straight to the inner ear through the bone. And it works very well. Many people use cochlear implants now with great success. Anyway, this is our very large ear model. So, and if you want to get an idea of how big it is, it's about 2 feet across. So anyway, get back to what I was talking about. Okay, so there's our little tiny one. So okay, now, so the inner ear has 3 parts. It has the semicircular canals, it has the vestibule, and it has the cochlea. The cochlea only deals with hearing. The vestibule and semicircular canals are related to balance. The semicircular canals move in 3 planes, a lateral plane, an anterior plane, and a posterior plane. And the vestibule then has hair cells in there too for balance and movement. Okay, so there's the semicircular canals right there. On the inside of the canals, we have these hair cells right there that vibrate. They vibrate in the direction that they're moving. As we turn our head, as we accelerate in a car, as we slow down in a car, those hair cells move. We know that when we're accelerating, we know when we're slowing down, we know when we go around a curve. I mean, obviously when you're driving, you know that, but your brain tells you that also because of the angular motion of these hair cells. You go to Dollywood and go on a ride in Dollywood, and you know, depending on the type of ride it is, you might shut your eyes to go on a ride. It happens. You might be too afraid to look, but you're going to know when you're moving in different directions because of the semicircular canals telling you what's going on. If you get off the ride and you're dizzy, it's because these hair cells need a little time to reset. You know, after going on the Eagle or some other ride that might take you upside down and around in very steep circles, your hair cells need a little bit of time before they come back to equilibrium. And so you might get off the ride and have a little trouble establishing your balance because of that. Okay. So as I said, the cochlea is the snail-shaped structure where our hair cells are located. They look something like this. These are hair cells here. These are found scattered throughout this region inside the cochlea, and they will vibrate when the pressure waves strike them. Now they function as mechanoreceptors, and they can generate graded potentials. And those graded potentials will reach the cell body and hit threshold if they can depolarize to -55. Just because they vibrate, excuse me, just because they vibrate doesn't mean we're going to hear a sound. Got to hit threshold, you know, so our ability to detect certain sounds and patterns of sound depend on these hair cells, these mechanoreceptors being able to reach threshold. If they don't, then we can't hear it. And that accounts for why we can hear things in the mid-range and not in the high or lows, or why we can hear the highs and lows and maybe not the mid-range, or maybe we can hear some of the highs and some of the lows and everything in the mid-range. It just depends on how effective our hair cells are in hitting threshold. So, yeah. And there are membranes where those are the hair cells right there. Now a test for conduction deafness. This is something that we do when we're always on the ground here. We take a tuning fork, and if you strike the tuning fork and press it against your mastoid, process, you can hear the note being played by the tuning fork. If you hold it against your mastoid process until the sound goes away, and then take it and hold the forks of the tuning fork against your pinna, like you see in picture number 2 there, you'll hear the sound again. So the way this works is when you hold it against the mastoid process, the vibrations from the tuning fork are passed directly into your bone, and they go straight to the inner ear, and you can hear the musical note that the tuning fork is playing until the mastoid process can't detect those vibrations anymore. However, tuning fork's not done vibrating. If you then take it and hold it against your ear, like you see in the second picture here, it's still vibrating, it's still generating a musical note. And now, if you can still hear that note from your external ear, it indicates there is no conduction deafness going on. You heard the sound 2 different ways. You bypassed the middle ear and went straight to the inner ear by pressing against the mastoid process, or you went through the auditory canal. And a very simple process here. Let's see if I can do this without sticking myself in the eye or something. Okay. Now, here is a tuning fork right here. And if I strike the tuning fork, everybody should be able to hear the sound of the tuning fork, right? Everybody should hear that. Okay. Now I'll strike it again. And if I place it against my mastoid process, right there, I can hear it very well. Everybody should be able to hear it. And it's starting to fade out. It's taking its good old time to fade out. Okay, now if I take the tuning fork and hold it against my pinna, you should be able to, you might still hear that, because I can hear it now at the pinna. So I don't know if you can hear it or not on the microphone, but I can, but the external ear is picking that up. So that's showing me that there is no conduction deafness going on, because I got it through the mastoid process. I got it through the external ear. And it's a very simple test for conduction deafness. If there's a problem with transmission at the middle ear, then a cochlear implant in the back of the head, in this region, usually right about here by the mastoid process, or a simple hearing aid can work. But if it is nerve deafness, none of those devices are going to help. So anyway, let's get back to this. Okay, now that's the tuning fork test, and it works very well. We do it all the time. So anyway, now let's move on and take a look at the eyeball of a sheep. So this is a sheep eye right here, and this is what you can see a lot of structures on this eye already. First of all, you can see that the dark area in the front of the eyeball is called the sclera. You know, the sclera is, in this case, it's a dark area because it's covering the iris and the pupil. Now, the typical sclera, though, is transparent. If we look at the model of the eye here, we can see there we go. There's our eyeball. This transparent area here, move this thing back a little bit. Let's see if that works. There we go. Okay, this transparent area here is the sclera. Well, I mean, the white area is the sclera, and this is the cornea right here. So that dark area you're seeing on the sheep eye is represented by this cornea right here. What's underneath the cornea is what's making it appear dark. So let's take a look here. So there's the cornea. There's a lot of fat around the eyeball for insulation and protection. Not that the sheep needs it anymore. There are a series of 6 muscles that surround the eyeball. These muscles allow our eye to move around when we're looking at things. These are these muscles then control the movement of the eye. The white portion around the eye, the outer covering of the eyeball, is called the sclera. And you see it on our model here. The sclera is going to cover every portion of the eye except where the cornea is located. So there's our cornea right here. And all of this is the sclera that we see here. So maybe on a smaller model, it might help. Let's take a look at this. This is our little eyeball. And so our sclera is this white area all the way around. Only the cornea is transparent. The rest of it is white, even if half of our cornea is missing on this model. So anyway. Okay, so there is our sclera. Now, if we flip our eyeball over, we can see where the optic nerve is located. The optic nerve is a structure about the size and shape of a pencil eraser. It has the texture of a pencil eraser too. It's very, very rigid structure. And that's the optic nerve right there, because this is a collection of lots and lots of axons. Remember that nerves are axons bound up in myelin that are outside the central nervous system. In the peripheral nervous system, axons covered by myelin are the nerves. Inside the central nervous system, they're called tracts. So what you're seeing here is the optic nerve. It comes out the back of the eyeball here, and it's surrounded by layers of fat. Now, when you have an eyeball in your hand and you put your finger down on the optic nerve while you're wearing gloves, you can tell that it's not just muscle or fat, because its texture, as I said, is very much like a pencil eraser. And it's about the size and shape of one too. So fat layers, eye muscles there. You can even see a little bit of the sclera down here in the side. So now there's the optic nerve right there. And there it is again over here. Again, you can push away the tissue around it and it will stand up, and it is very firm. It is very much like an eraser. So optic nerve there. There's the cornea again. Now the cornea, and I'll show you in a little bit here that the cornea really is transparent. So sclera. There's, oh, I got carried away. There's the optic nerve again. Again, now with some of the soft tissue pushed away from it. And nerves are like this. Nerves are very, very rigid structures. They're very strong. You know, they're not like muscle. They're not like bone. They're not like connective tissue. They are a separate structure, and they are very, very firm. So, okay, now, if we take our eyeball and we dissect, make a cut in the eyeball where we cut, we make a longitudinal cut in the eyeball. So what we've done here is we've exposed the inside region of the eye. We've taken that eyeball and we've essentially made a cut straight up and down in this essentially you're looking at the results of a sagittal cut, slightly off center because the lens is completely intact. but this is still what we call a sagittal cut because we're dividing it into a left and right segment. So you can see a lot of structures here on this cut. First of all, there's the cornea right there. This opening here, this jagged edge here, this is part of the pupil. The pupil controls light coming into the eye. In this area that forms the pupil is called the iris. That is the pigmented portion of the eyeball. Now, in front of the iris and behind the cornea is what's called the anterior chamber, filled with fluid, salt water, lacrimal fluid. You know, when that pressure, if that fluid doesn't drain properly, and we use that fluid to wash our eyeballs off with when we blink. If we don't drain that fluid off, then we start putting pressure on the retina and start destroying the retina. And that, of course, is the condition known as glaucoma. Now here is the vitreous chamber or the posterior chamber. The lens divides the eyeball into an anterior chamber and a posterior chamber. Now the posterior chamber or vitreous chamber is filled with jelly to give the eyeball its shape. And of course, it's a lens right in the middle there, about the size of a large M&M or maybe a Skittle. You know, it's sort of it's round. It's made out of a crystalline protein. It's transparent. It doesn't look like it now, but it really is transparent. There's our sclera again on the outer covering of the eye. Oops. There's our cornea again. This structure right here is the pupil, and the pupil is surrounded by the iris. Now, these structures here in black are the ciliary bodies. Those are muscles which will cause the shape of the lens to be bent or to condense or to spread out. They allow the lens to accommodate objects that are up close or far away. We can change these black bands of muscles here allow us to change the shape of our lens so that we can look at something up close and then immediately go look at something far away without having to stop and think about it. Okay, so there's our lens. We're looking straight down into the eyeball. Now, this kind of cut has been, this is a transverse or horizontal cut looking straight into the eyeball. There's the lens looking down into it. This black area here is the ciliary body. Those are smooth muscles that will change the shape of the lens as we need to. And then just see the fringe of the sclera here, and this light brown area back in here is just the tips of the retina. Okay, so lens, sclera, ciliary body. Again, they will allow the lens to change its shape, will allow the lens to change its shape to accommodate objects that are up close or far away. Yeah. Now this is the, we made a cross-sectional cut of the eyeball right at the level of the lens. Now this is the portion that we removed to get this picture here. We had to take off the front of the eye, and that's what this is. Now, if you think the cornea wasn't transparent, you're looking through the pupil from the inside. That gray structure you see in there is actually the front of the eyeball. You're looking through the cornea. So this is the iris, right here. This is the pupil. This is the ciliary body. On the other side of that is the iris. So, and you're looking at it from the position of the lens right now. Let's see what we've got labeled here. Ciliary body, pupil, sclera, and now we can see the iris. This brown area here, these two layers, you can see the muscles run in two distinct layers, longitudinally and circular. And this is the pigmented portion of the eye. Our sheep had brown eyes, and the iris controls the shape of the pupil to allow light to come in, to restrict entry of light in bright light, and to allow more light to come in dim light. And this is the iris itself. This is what corresponds to the pigmented portion of our eyes, whether it's blue or green or brown or black or hazel or gray or whatever. It's the iris and the pupil. Let me go back. Okay, now this next slide coming up shows what happens if you open up the vitreous chamber, the back portion of the eye. As I said last week, that it's filled with jelly. And because it's filled with jelly, it can give the eyeball its round shape. The jelly is optically transparent. Light rays can pass through that and strike the retina. And we call this the vitreous humor right there. And so if you take it out of the eyeball completely, it comes out in sort of a round mass. And that's what you see right here. And over here we see, we put it on the dissecting tray. There's the vitreous humor here. It just sort of collapses onto itself. There's the lens. These are the suspensory ligaments here that hold the lens in place. And we just sort of see part of the ciliary body left dangling on the vitreous humor. See, and you can see it up here because there's the tip of the lens right there. And there's the ciliary body surrounding the lens. And can't sort of make out. You can on this one, you can't hear it all. But these are the suspensory ligaments. Okay, so let's take a look at the back wall of the eyeball. So first of all, we've got, and this is a very nice picture here. We have the lens, we have the ciliary body surrounding the lens, we have the vitreous humor. So again, it's a ball of jelly, which is optically transparent. Light rays come in through the lens and go through that vitreous humor to strike the retina. Over here is the retina. The retina is very, very, very thin. This whole thing is the retina. Where the blind spot is located is where the optic nerve is leaving the eyeball. And so no light rays that strike there aren't going to be detected by anything. But the thickness of the retina is probably less than a millimeter in thickness, maybe in tenths of a millimeter. It's like the equivalent of a piece of single-ply toilet paper laying on. It forms the inner surface of the retina, and so you can see the structures here in the retina that all lead. All what you're seeing are the axons from the ganglia, all leading to the blind spot. Just to give you an idea of how thin the retina is, this is also the retina here. It's been torn a little bit, but it is extremely thin. Again, there's the blind spot right there. Now, this blue-green area is something that animals have that humans don't. This is their night vision device. Animals have this iridescent blue coating on the surface of the choroid coat. The choroid coat is the middle layer of the eyeball. You have the retina, you have the choroid coat, and then you have the sclera. Now, animals have a tapetum lucidum located on top of their choroid coat. This collects and amplifies light at night so that animals have great night vision. This is why animals' eyes will glow in the dark when light strikes them. You can see a deer at night, you can see a possum, you can see a fox, you can see cows, their eyes glow. You're out in the yard at night with your dogs, for example, and your dog's eyes glow because they have this tapetum lucidum. We don't have that. So our night vision suffers accordingly. Animals have great night vision. So we don't have that. It's very pretty. It's iridescent blue. We don't have it. There's the choroid coat, the dark layer here. It's dark, designed to absorb any extraneous light that bounces into the eye. So we want to keep everything focused on the retina. There's the retina. Again, you can just see just how thin that layer is, where the photoreceptors are located. Photoreceptors cover this whole structure, and everything leads to the blind spot right there where the optic nerve is leaving the retina. And this is the optic nerve, by the way, over here coming off. Now, and usually when we're dissecting, if someone dissects out an eye this well, that's pretty good. Most people end up, when they pull the vitreous humor out, the retina comes with it, and it can't be helped. So, but these are the structures of the eyeball. Now, I want to emphasize again back here in this other picture, that this cross-section picture right here, this sagittal section, gives you a great look at half of the pupil right there. And then this is the iris located here, just to remind you of that. So, okay, let's go on and take a look at the brain. Now, this is a sheep brain. The sheep brain is about 1/4 the size of a human brain. It has many of the same structures, so it's a pretty good comparison. The lobes of the brain is made up of a series of lobes that share the same name as the bone that sits on top of them. So, for example, the frontal lobe is covered by the frontal bone, the parietal lobe covered by the parietal bone. The occipital lobe, located at the base of the brain, is covered by the occipital bone. And we have the temporal lobe on the sides covered by the temporal bone. So here is our brain. Now, we notice right away there's a big groove down the middle. That groove is called the longitudinal fissure. And that divides these are the cerebral hemispheres we're looking at, the two largest portions of the brain, the left and right cerebral hemisphere, separated by this longitudinal fissure. And so on a human brain, it would look something like this. This is here is our life-size brain model right here. And this is the longitudinal fissure right here. This is a groove that separates the two cerebral hemispheres here and here. And so we have there's, it's not unusual to have grooves like that. You can see there's a groove here, and there's a groove here to separate. This is the occipital lobe back here. This is the temporal lobe off to the side. There is the parietal lobe here. And so it's not, these grooves are not unusual. So what we do have is the frontal lobe, the parietal lobe, frontal here in the front. There's the frontal lobe, the parietal lobe, the temporal lobe at the side, and the occipital lobe back here, on this portion of the brain, here and here. Now, directly below that is the cerebellum. These two areas here are the paired cerebellum. They're the second largest portion of the brain. The cerebral hemispheres are the largest, the cerebellum is the second largest. And the spinal cord comes down, is present right here, where it connects with the midbrain. Midbrain is, I'm sorry, with the brainstem. The brainstem is up inside here. So anyway, that's how big the human brain is. And the sheep brain is about half the size of the human brain. So there's our longitudinal fissure. We have a series of ridges and grooves in our brain. And those ridges and grooves are designed to increase surface area. Cerebral hemispheres, yeah. The ridges are called gyruses, and the grooves are called sulci. Like there's the cerebellum, there's the spinal cord, there's a sulcus. A sulcus or a sulci is a groove. You know, we use the grooves a lot of times as landmarks. The ridge or the bump is called the gyrus. There's a gyrus right there. And so our brain is covered with sulci and gyruses or gyri. What they're doing is increasing the surface area of the brain itself. Frontal lobe, oops, let me go back. Frontal lobe, occipital lobe, and parietal lobe right here. Temporal lobes on the side, you can't see it very well on this. So, cerebellum, cerebral hemispheres, gyrus, sulcus, longitudinal fissure, occipital lobe, parietal lobe, and frontal lobe, and the spinal cord down here. Okay. Now, if we flip our brain over and look at the underside here, at the ventral surface, we can see spinal cord. This is the medulla oblongata. This is the end of the brainstem. Where the spinal cord ends and the medulla oblongata begins is problematic, but we see that they tend to run together here, somewhere below the foramen magnum. There's the pons. The pons here, the pons is always a crossing over region where the spinal nerves that are coming up as tracts in the white matter of the spinal cord cross over. See, because the brain has contralateral control, which is a fancy way of saying the left side of the brain controls the right side of the body, and the right side of the brain controls the left side of the body. The crossover of the nerves is taking place at the pons. So nerves coming up as tracts on the left side of the spinal cord cross over to the right side of the brain, and tracts on the right side cross over to the left side of the brain. Now, this structure here is the pituitary gland. It's like it will sit in the sella turcica of the skull of the sheep. Pituitary gland in a human is considered the master gland of the entire body. It releases it releases 8 different hormones to affect different things in our bodies. We'll talk all about those hormones when we get into AP2, when we talk about the endocrine system. Now here in the side, the temporal lobe is exposed now. It's underneath the temporal bone. Now this structure here is the optic nerve. It's not a tract yet because it hasn't entered into the brain. The optic nerve is one of the 12 cranial nerves. Remember, it's in the peripheral nervous system. It's not in the central nervous system. That's why it's a nerve, not a tract. It connects to the eyeball, one eyeball on each side, and the optic nerves then cross over right here at this structure called the optic chiasma. And that crossing over allows the optic nerve to send a branch to both the left and right occipital lobe. So each optic nerve coming off the eyeball, there's 2 optic nerves, 2 eyeballs, 2 optic nerves. The optic nerve each has 2 branches, and they send them back. Each branch goes back to the occipital lobe. The occipital lobe on the left and the occipital lobe on the right. So what we end up with is binocular vision, so that both eyeballs are sending 2 signals to the occipital lobe. Each eyeball is sending a signal to the right and left occipital lobe, which puts that image together into a 3-dimensional binocular vision. and the optic chiasma is the crossing over point. Now this structure is known as the olfactory bulb. The olfactory nerves come up out of the nasal cavity through the cribriform plate, through all the perforations in the cribriform plate, and enter the olfactory bulb, and then leave the bulb through the olfactory tract, because now you're in the central nervous system, to the temporal lobe, so we can tell what we're smelling. So there's the olfactory bulb right there, one on each side. In the sheep, these are massive compared to what ours are like. Here's another, we have removed, if you remember this picture back here, had a lot of connective tissue on it. Those are the meninges. That's the dura mater right there. We talked about the meninges in lecture today. Now, if we take a look here, most of the meninges have been removed and pushed back. And we can see them, again, this is the dura mater layer here. Now, the shiny layer on the brain, and you can just see part of it right there and there. There's a little, that membrane, that is the pia mater. Arachnoid layer is just sort of not visible here. So, okay, let's take a close look. There is the spinal cord. There are the meninges. This is tough connective tissue. There's the pia mater, all this shiny stuff on the surface, you know, just sort of the material here and here, just adhering to the surface of the brain. That is the innermost layer of the meninges. Pituitary gland again. Temporal lobe. What's left of the optic chiasma, in this case, most of the optic nerve on this particular brain has been cut away. Why? Don't know. Sometimes they come that way. Now here's your olfactory bulb. Now coming from each olfactory bulb is the olfactory tract. This is a tract right here. We're now inside the central nervous system, so it's a tract and not a nerve. There's an olfactory tract there and one down here. Now, every sensory input that comes into the brain passes into a structure known as the thalamus. The thalamus is part of the diencephalon. The top, you know, it sort of caps the brainstem, and everything goes into the diencephalon and gets routed to different parts of the brain except odors. Odors get passed to the mammillary body and then to the hypothalamus. The hypothalamus controls odors. So now let's go. So there's the mammillary body right there. Let's take another look. There's the dura mater again on the meninges. We're still looking at the underside of the brain. Again, there's the spinal cord. There's the medulla oblongata. That is a key, that is the lowest portion of the brainstem, but it is a key area of reflex and control for us. There's the pons. Again, that's the crossing over site. Trigeminal nerve, or what's left of it. The trigeminal nerve is a cranial nerve. It has three parts to it. It innervates the area around the eye. It innervates the maxilla. It innervates the mandible. When we have dental work done on our maxilla or our mandible on either side, we will have the appropriate branch of the trigeminal nerve anesthetized with Novocaine or lidocaine to block all the sodium channels, so we don't generate action potentials and feel pain, because we already are. The pain receptors are still firing. You know, an anesthetic doesn't stop the pain receptors from firing. It blocks the signal. And so your pain receptors are still generating pain signals. And when those pain signals, those action potentials are moving up the trigeminal nerve, they hit the area where the lidocaine is and the sodium channels are blocked and the signal doesn't go any further. So we don't feel anything. At least I hope we don't. Anyway, okay. Mammillary body again. Dura mater again, right there. Temporal lobes on the sides here, you can see that. And here you can see the olfactory bulb. And here's a real good picture of the olfactory tract right there. So now this picture gets into something I was talking about today in lecture. The corpora quadrigemina, the reflex center for sound and for light. We have 4 protrusions here on the dorsal surface of the midbrain. This is the dorsal midbrain, and it's directly below the occipital lobe of the cerebral hemispheres. And you can see 2 large ones and 2 small ones. You also see a structure here known as the pineal gland. Now, there's the occipital lobe right there, one on each side. There's the pineal gland. The pineal gland secretes melatonin, not melanin, like we were talking about with the basal ganglia, but melatonin, which regulates our day-night cycle. So the pineal gland secretes melatonin. We get sleepy when it gets dark, and we're awake when it's light, all because of this. So it controls our, what we call our circadian rhythm or our day-night cycle. Directly below the pineal body is 2 large bumps, if you will, and that's the superior colliculus of the corpora quadrigemina. Superior because it's on top. Superior colliculus is the reflex center for vision. Sudden movement in your peripheral vision will trigger your head to turn in the direction of the movement. A bright light flashing off to the side, a sudden light, any kind of action will attract your attention here. Throwing something at you, throwing a ball at you. You can see the ball, track the ball, and know when to raise your hand to catch it. The superior colliculus of the corpora quadrigemina is enabling you to do that. It functions as a very important visual reflex center. Directly below it is the inferior colliculus of the corpora quadrigemina. This responds to sound. When we hear a noise, we turn our head in the direction of the sound. When someone calls our name, we turn our head in the direction of the sound. And when we hear an ambulance siren or a police car, or someone beeps their horn, or any noise, we turn in the direction of the noise or the stimulus in here. So this is the inferior colliculus of the corpora quadrigemina right here. Of course, there's the cerebellum below that. So this is on the dorsal surface of the midbrain. On the other side of the midbrain is that bird-shaped structure where we find the substantia nigra and the red nucleus, where we have the problem where dopamine production by the substantia nigra, or the lack of it, is the root cause of Parkinson's disease. So, okay, we have a sagittal cut of our brain. We can see a lot of structures on here. Right there is the optic chiasma, or what's left of it. We can see the meninges around the edge of the entire brain. Here's our pituitary gland. There is one of the cerebral hemispheres, or half of it, because the cut looks like it was made right down the middle on the longitudinal fissure. There's the cerebral hemisphere right there. There's the dura mater on the outside covering of the brain. This area here, called the corpus callosum, is actually a series of fibers that extend all the way into the cerebral hemisphere on both sides. And what we're seeing here is just the edge of it in the middle, but it actually extends out in both directions. The corpus callosum functions as a communication device to send signals from one hemisphere to the other and back and forth. This area back here is the cerebellum. And in the middle of the cerebellum is a structure here called the arbor vitae, Latin for tree of life. And this is the white matter in the middle of the cerebellum. White matter because it's myelinated axons. So there's the thalamus. The thalamus are paired thalami structures about the size and shape of a small egg, sits on the top of the brainstem. Thalamus receives all sensory input to get routed out to the various processing centers in the brain, except for odors. Looking at the outside of the same brain, we see the olfactory bulb. You can see the olfactory tract is right here. There's the pons, the medulla oblongata, the spinal cord, the cerebellum, the cerebral hemisphere. You can really see the gyruses, the ridges, and the sulcus, the grooves here. You can see some of the larger sulci here, separating the frontal lobe from the parietal lobe. Okay, that groove here in the human brain, we call that the central sulcus right here. Now, another cross section here, sagittal section, if you will. The spinal cord, the pons. I'm sorry, the medulla oblongata and the pons. The thalamus, better picture of the thalamus. This area up here is the corpora quadrigemina. There's the superior colliculus. There's the inferior colliculus right here. There's the corpus callosum. Now, inside the corpus callosum is a cavity. You know, we actually have a cavity. One of our fluid-filled cavities is inside the corpus callosum. Area called the fornix. There's the pineal body right there, this little structure right there. And then we have the superior colliculus and the inferior colliculus there and there. Now the openings in the brain are called the ventricles or aqueducts. Here is a lateral ventricle right above, surrounding the thalamus. There is the lateral ventricle in between, separating the corpus callosum. The third ventricle surrounds the thalamus. The central canal is this tube that runs the length of the spinal cord so that cerebrospinal fluid can pass up, down the spinal cord and back up. This is the cerebral aqueduct, this area directly underneath the corpora quadrigemina. The fourth ventricle is this area underneath the cerebellum. So the cerebral aqueduct connects the fourth ventricle to the third ventricle. The third ventricle surrounds the thalamus. The lateral ventricle is between the corpus callosums. And we have lots of holes in our head and filled with fluid. And then there's the central canal there. So, okay. And that is our eyeball. I mean, it's our brain. Any questions about any of this? So those are the structures then of the ear, the brain, and the eyeball. So, and of course, all these PowerPoints are in your content section too. So, any questions? Okay. Well, you might want to take a minute to reflect that this is the last lab of AP1 Lab. So anyway, let me get us out of here. I need to fix those tests so that you can see them. And I'm not going to open the lab exams because you're not going to have a final exam in lab. So, and I'm also planning, if we get done with lecture in time, and I'm trying very hard to do that The last day of class for both AP1s and the AP2 class are going to be review days for the final exam. So in your case, it would be the, let's see, it would be the 27th of April, and for the Thursday class, it'll be the 28th of April. So hopefully we'll have all the lecture content done so we can use that last day of class for review. That's my plan anyway. And I will tell you that people that go through the final exam review do a whole lot better in the final exam than just going in cold. So not that I give you any answers. I wouldn't do that, but it just makes you think. So next Monday for lab, we have no lab next Monday. We're done. I will get around to posting the lab test, the fourth lab test, probably in the next week or so after I write it. But we are done with lab for the semester. So. Any questions? Okay, then I'm going to get us out of here so I can get some stuff fixed for you. So, and I will see you in lecture on Wednesday.